Biology. This recording, we're going to go over the lesson gene expression and gene regulation. And I'm going to apologize. This is another long one. There's just a ton of information in this lesson. It really should be more than one lesson. Remember that if there's a yellow star in the left-hand corner, that means a quick check question is being answered. Okay, so first off, gene expression. Let's review what that means. So a gene is a segment of DNA that codes for a protein. So not all of your DNA makes proteins, but the parts that do are referred to as genes, and those proteins do something in your body. For example, creating your eye color. Your body is making certain proteins and different amounts of them to help create your eye color, and that's why a lot of us have different colored eyes, is because we're making different amounts or even different types of proteins. Now, gene expression, for a gene to be expressed, for it to be turned on, it means it has to be undergoing transcription, and that mRNA has to be translated into a protein. So for a gene to be expressed, both of those processes, transcription and translation, need to occur. Now, gene regulation is all about controlling gene expression, right? Because every cell in our body has the same DNA. And we need those cells to be specialized and do specific tasks so that way our body can function, right? So all the cells in our body have the same DNA. What makes them different? What makes your bone cells different from your blood cells, different from your muscle cells, are the genes that are turned on or off. So gene regulation is about controlling gene expression, turning some genes on, turning some genes off. So if we're turning a gene off, that means transcription and translation are not happening. If we're turning it on, then those processes are happening. Now, one thing that makes this lesson so long is that we're trying to cover these processes for both prokaryotes and eukaryotes. So remember, a prokaryote is your single-celled, smaller organism. It doesn't have a nucleus, it doesn't have any membrane-bound organelles. They're one cell big, where eukaryotes are very complex. They have a nucleus, have these complex organelles, and many of them, like ourselves, are multicellular, meaning that different cells are going to have different functions. So regulation is super important for them too. Now, for both types of organisms, prokaryotes and eukaryotes, you can control this process at different levels. You can control it at the transcription step, which your lesson includes the DNA packaging and chromatin remodeling as part of its own thing, but I would argue that it's part of transcriptional control. Eukaryotes have this extra step, extra place. We can control gene expression at the post-transcriptional level, and then both have a translational control, and then both also have post-translational control. All right, so let's start looking at how we can control these processes for the different organisms. First, we're going to begin with a refresher on transcription. So remember, for transcription, you're going from DNA to RNA, we're the same language. The enzyme that's involved is RNA polymerase, and it's the one that copies the DNA to the RNA, and they bind to a promoter. So there's a certain sequence on the DNA referred to as the promoter. That's where the polymerase binds, and it'll start making the complementary strand. It uses a template of DNA to make a complementary strand. That happens in both prokaryotes and eukaryotes. They have this enzyme, the polymerase, and the polymerase has to bind to a promoter. Okay, so gene regulation for prokaryotes. Prokaryotes have one chromosome, one circular chromosome, not in a nucleus. And one thing I love about prokaryotes is that they're organized. Their genes are organized into operons. So this term only applies to prokaryotes. We don't have this. Prokaryotes have operons, and an operon is a promoter, so where RNA polymerase binds. It has an operator, and it will have all the genes needed for a certain chemical pathway. I love it. So all the genes needed for a certain chemical pathway are all organized together, all regulated by the same promoter. It's very simple and clean, I think. And from this picture here, you can see we've got this regulatory gene and promoter for the regulatory gene, and then the actual regulatory gene. So all operons have a regulatory gene, and they're referred to as repressors. So repressors are the regulatory genes, and they bind to an operator and turn off the operon. So in my picture here, I've got, those regulatory genes. They're not necessarily right next to the operon. They can be someplace else. Just the image is showing it's there. Then for the operon, we've got the promoter, where polymerase binds. The operator, the operator, which is where the repressor binds. And then all of the genes needed for that chemical pathway. In this case, it's the lac operon, which all the enzymes we need to digest, or not we, but a bacteria needs to digest lactose, which is a type of sugar. So the repressor, if it's bound to the operator, then this whole system is turned off. If the repressor is bound, then the polymerase cannot transcribe these genes. Something has to inactivate the repressor and allow polymerase to move forward. Right? There's different types of operons, and there's a couple videos in the lesson that you can watch about the lac operon and trip operon, classic examples. But that's the basics of controlling transcription for a prokaryote with that regulatory protein called the repressor and their operons. Now, eukaryotes, like ourselves, we can control transcription by reorganizing our chromatin. So the chromatin remodeling is how tightly wrapped the DNA is around the proteins. Because remember, chromatin is DNA and protein wrapped around. So in this picture right here, we're starting to get to the chromatin, where the DNA is wrapped around those histone proteins. And there's different things that can happen to the DNA that change how tightly wrapped it is. The tighter wrapped, if it's really tight, like in this picture, the promoter, the polymerase can't get to the promoter. There's no space for it to get in here, right? So if it's wrapped up really tight, it's almost turned off completely. Or there's different things that could happen to these proteins that cause them to be further apart, and now there's space for the polymerase to come in and transcribe the DNA. So that's one way of controlling transcription, is giving access to the promoter. Another way is through regulatory proteins. So prokaryotes have regulatory proteins, the repressor. Eukaryotes do too. We don't call them a repressor, they're called transcription factors. They are factors that help either enhance or inhibit transcription. There's different types of them. But they help control gene expression by affecting transcription. So again, with eukaryotes, each gene has its own promoter and their own transcription factors. All right, with eukaryotes, and again, this is a step that prokaryotes don't have. They don't have this step, so there is no post-transcriptional control, which is RNA processing. So RNA processing is only in eukaryotes. And this is when the RNA that gets transcribed is changed to create this mature mRNA. So we have transcription, and there's a processing, there's RNA splicing that happens where certain segments of the RNA are removed, and then the parts that are, were cut out are resealed together, and now you have this mature mRNA. There's some other things that happen with the 5G cap and the 3' poly A tail. But I would focus on these introns and exons. I wish the exons exited and the introns stayed in. That would be lovely if it worked out that way, but it doesn't. So how I remember exons and introns is that the exons get to exit the nucleus. So again, we're talking about a eukaryote. This doesn't happen in prokaryotes, just eukaryotes. We've got our DNA that gets transcribed into RNA. Then that RNA gets processed. Part of that processing is splicing, RNA splicing, where the introns are removed and the exons are kept in there, and they get to exit the nucleus and go into the cytoplasm where translation occurs. So exons get to exit. They stay part of the mRNA. Now here's something that's kind of cool, that's higher level, but different cells can process this mRNA differently. So different cells, different parts are going to be introns and different parts are going to be exons. So from the same transcript, you can actually get different proteins, okay? If there was a mistake that was made and they left introns in, then they'd be there for translation and you would have extra amino acids and it probably wouldn't work. So again, RNA processing, this is a post-transcriptional, that should be changed, my bad, control, and it's just seen in eukaryotes. All right, so translational control. So remember, translation is going from mRNA to a protein. So with translational control, we're trying to control how that happens. And this step occurs in both prokaryotes and eukaryotes once it's transcribed. Both of them have things that go around and destroy mRNA. If it's destroyed, then it's not there to be translated. There's also RNA interferences, these little molecules that can go in and silence the mRNA. It doesn't destroy them, so they're still there, but if they're silenced, they're not being translated. Another thing that could happen is changing the amount of ribosomes that can attach. You can have more than one ribosome attached to it. The more you have, the more protein that gets made. And the more protein that can change the expression, what's happening. So different steps that can happen with translational control. And then post-translational control, again, this is both prokaryotes and eukaryotes. It controls the protein after it's produced. So anything that regulates the protein, how it's functioning after it's produced would be a post-translational control. It could be how long the protein lasts. Some get degraded really quickly, some last a long time, or maybe if it's active after translation, sometimes that protein needs another protein or another something to make it active. So any of those would be under the umbrella of post-translational control, and it can be seen in both prokaryotes and eukaryotes. All right, your multiple choice question is which is only seen in eukaryotes? Was it transcriptional control, RNA processing, translational control, or post-translational control? And the answer is your code word. But before you go, one thing I want to cover, because it's a question on the quick check, and I just want to make sure that you have hear about it before, but about Hox genes. So Hox genes are a series of genes, or a bunch of genes that code for transcription factors that regulate body development. So remember, transcription factors are those regulatory proteins in eukaryotes that encourage, or they could permit or stop transcription from happening. And this series of genes are specifically regulating body development. So what's going to develop into hands, arms, the midsection, the legs, etc. Now there's been lots of studies on Hox genes using mice, and what they do is they cause them to become mutated and see what happens. So one of the examples is they've mutated a Hox gene, and what happened was they had the flies would have double wing development, okay, which flies don't usually have. Another kind of crazy mutation that they've seen is with the antenna. Instead of developing into antenna, they develop into legs. So the wrong genes were turned on, the wrong transcription factors were made, and the wrong genes turned on, and instead of developing into antenna, it developed into legs. So multicellular organisms have the same Hox genes. We all have the same ones. There's some that might be slightly different. Different ones are turned on and off. But the fact that we all have this group of genes shows that body plans of complex animals are very similar to one another. And that's the answer to the quick check question. All right, we made it to the end. Thank you for watching, and let me know if you have any questions.